All right, welcome back to Life and Biology. We're going to go over the next category of macromolecules in the class, and that will be lipids. But just to finish up the polysaccharide section, I wanted to mention that even though we're not going to dig deep into drawing out all sorts of polysaccharides, that there are some functions for polysaccharides in living systems. Sometimes the purpose is for energy storage, and other times the purpose is for structure, and there could even be more purposes. Decorating proteins, for example, with sugar moieties. Anyway, plants use, to store energy, plants often use starch. So in the lab this week, we're doing a test for starch, and when we drop iodine on a potato, it turns black right away because it's interacting with the starch molecules. And then another very common polysaccharide that you would find in plants is cellulose, and the purpose of cellulose is for structure. Next, we've got in animals, they don't necessarily use starch for energy storage. It's more often glycogen. And glycogen, when glucose enters the cell may not need to break down the glucose right away. And so rather than going straight into the cellular respiration pathway that we're going to discuss later in this semester, the glucoses can be linked together into glycogen molecules, and it's at a fairly compact, easy to get to form when they do that. So that's an energy storage strategy in animals. If you start playing basketball or start running, your blood glucose will drop. And if that happens, then you can rely on glycogen in your cells for a while to provide the glucose that already within the cell. Okay, next up we've got chitin, and this is something you'd find on arthropods. And chitin arthropods are things like insects and lobsters and anything with a shell or an exoskeleton. And you might have learned about that in your biodiversity class last fall. But chitin is going to be a very strong structure, exoskeleton. That's the outer skeleton on these kinds of organisms. Okay, that's the last. We'll talk about polysaccharides for a while. We're going to switch over to lipids now. Apologize, this slide is a little bit hectic. So in class, I was like drawing these red lines to try to separate out my different parts. So we'll talk through this slide in the order we did in class. Now, lipids have regions that look like hydrocarbons. And when we learned about hydrocarbons, we learned that when you have a bunch of hydrocarbons in water, they'll stick together because they are nonpolar. They're hydrophobic. They don't like interacting with water. And so they will form clumps together. The shortest hydrocarbon we learned about was methane. It has just a single carbon. And it's nonpolar. Then we have ethane, 2 carbons. There's a single bond, and then you fill out the rest with hydrogen. So C2H6 is ethane. And the thing I wanted to point out here is that the 2 carbons that you have here can rotate around each other, or they can rotate around this covalent bond here. Whereas in ethene, when there's a double bond created, the hydrogens won't form a double bond. So if there's a double bond, it has to be between the carbons. You lose a couple hydrogens because with 2 bonds between the carbons, you only need 2 more bonds to satisfy the octet rule for these carbons, the 8 electron in the outer shell rule. And so now we have just 2. But with this double bond, there's some constraints. This covalent bond allowed rotation around the 2 carbons. When you form a covalent or a double bond, these can't rotate. And so this molecule is stuck in kind of a flat linear plane. These 3 hydrogens can rotate around in 3 dimensions. Okay, so if we draw a long hydrocarbon, it gets tedious to draw out all the carbons and hydrogens. So we just draw a zigzag line. Anywhere the line bends or ends, you have a carbon. So the bends and the ends are a carbon. And this one in particular is drawn without any double bonds. And so this molecule here is very flexible. And you can pack them in tight if you want and that sort of thing. Whereas this molecule here, I have drawn some double bonds on a few occasions. And so even though it's still a long carbon chain, each time you put a double bond within a hydrocarbon, you're going to reduce the flexibility of that. Now, I drew the double bond here where you have a zig and a zag, is how I describe that kind of Z pattern, a zig and a zag. Here, I've drawn a double bond where you do a zig followed by another zig before you go back to zigging and zagging. And to use more scientific terms, this switching pattern right here that I'm indicating with my obnoxiously large cursor is called a trans situation. Trans is the conformation around that double bond, whereas this double bond where they have the R groups are both going off in the same direction, that's called cis. Just to drive that point home a little bit better in class, we drew this out where rather than having a whole carbon chain there, I just wrong direction. I just used R1 and R2 as R groups that can indicate that this just goes on for a long time. So here's we've got our carbon double bonded to another carbon. Use some hydrogens just to fill that out. But we got R1 going up, R2 going down. So that is the trans conformation. Whereas here's a carbon double bond. Both R groups are pointing down, and that's cis. So it's a lot the same as the structures that I drew up here, but it's just isolating it down to the most simplified way of describing cis and trans conformation. Why am I bringing up cis and trans? It's because in nature, you tend to have a lot of cis carbons, and trans are something that happen in a food processing step. So if you hydrogenate some oil, you will sometimes cause trans bonds to form. So then you get trans fats. And in the, let's see. Okay, yeah, I'll finish up this discussion here before I move on to the next slide. In the 1980s, when I was a kid, people would eat a lot of margarine or oleo, and butter was considered unhealthy. Well, the margarine and oleo and other things have this trans conformation. It's an unnatural conformation, and it turns out that unnatural fats were actually not good for your heart. Whereas butter will tend to have the cis conformation, and that is, even though that was considered the unhealthy version back in the 80s, we now know that this is far more healthy to consume than fats that have these trans bonds in them. Okay, so lipids, taking a zoom back out to lipids, we were just talking about hydrocarbons for a long time, but these are nonpolar for large segments, and it's nonpolar for those regions where it's the hydrogen and carbons that make them up. We're going to cover 4 categories of lipids: fat, oil, waxes, and I had written cholesterol, but the more general term is steroids. We're going to draw out a molecule of cholesterol, and that's why I had screwed up or switched or accidentally said cholesterol was the 4th group. So steroids is the general name for this entire group, and cholesterol is one type of steroid. I'll start with fat and oil. These have very similar structures. One of the differences is that fat is solid at room temperature and oil is liquid at room temperature. And so what we have going on here is that there are different numbers of those double bonds that can lead to either fats or oils. But in the long run, each fat or oil molecule has a glycerol backbone and then 1 to 3 fatty acids. So to draw that out here, I have blue indicating the glycerol backbone. Glycerol is a 3-carbon sugar, which means it's a sugar. Sugars are actually alcohols. The OH group is here, and so you can see the OL here for alcohol. So we take a 3-carbon sugar and put some hydroxyl group on it, and it turns into glycerol. Okay, so the 3 hydroxide hydroxyl groups that are pointing out from the glycerol can be lined up in such a way that the fatty acids, the 1 to 3 fatty acids, are pointing in one direction. And in this case, where there's 3 of them, we're going to call it a tri for 3. Acyl, referring to the acyl chains. That's what you call these fatty acid, the carbon and hydrogen portion of a fatty acid. And glyceride, because it had the glycerol backbone. So once it's formed those bonds, it's a glyceride. So triacyl glyceride, or you might shorten that up to TAG. If you only have 2 fatty acids, shown here, then this third one still has a hydroxyl group or some other thing attached to it, but 2 of the hydroxyl groups have a fatty acid that have been put onto those carbons where there was originally a hydroxyl group. Those are called diacylglyceride because di for 2, and so we could shorten that up to be called a DAG. And then finally, a monoacyl glyceride would have 1 fatty acid. I draw them this way because that way it looks like a capital letters, so that you could remember that the tri, di, and mono glycerides look like a capital E, capital F, and capital L. And just because I drew the acyl chain here doesn't mean it couldn't be on the top one or whatnot, but I just did that to make them look like capital letters. And then if we zoomed in on this triacylglyceride, again, we've got a glycerol backbone, and then you have 3 fatty acids connected to that glycerol. And at that point, it's a glyceride. So I should have probably written glyceride here. So just make note of that. Let's see. Okay, this is another messy slide. So let me just go through it in the order that I drew it out in class. We have fats and oils. These are glycerol and fatty acid, and they form an ester bond. So when a fatty acid sticks onto a glycerol molecule, you end up with an ester bond. So let's see what's going on here. Well, let's take a look. Let's start with the glycerol. Here's where I drew out the 3 different carbons, and then I have the 3 hydroxyl groups, and then we will know that those oxygens are electronegative, and so that's kind of messy looking, but that's a delta minus, so it means a partial negative charge there, which gives a partial positive charge on that carbon. Let's take a look at a fatty acid. A fatty acid looks like this, where you've got your zigzag of carbon and hydrogens, your hydrocarbon region. Then you've got a carbon, and the end carbon has a carboxyl group. So a carboxyl group has a double bonded oxygen as well as an oxygen and a hydrogen. So it's a carbonyl and a hydroxyl, and you combine those together, you get carboxyl. Carbonyl plus hydroxyl equals carboxyl. And so this one is, we learned that this one might want to be a negative charge when we learned about functional groups. So at physiological pH, which is a pH of roughly 7.4, typically in the cytoplasm of a cell, it would have a charge of -1. Why does it have a charge of -1? It's because this oxygen was greedy. It pulled the electrons completely away from this hydrogen. A hydrogen without its electron is just a proton that floats off. Any molecule that produces or is willing to give up a proton is an acid. So we got the fatty part, the hydrocarbon part is the fatty acid or acyl chain, and then we've got a carboxyl group here, which is an acid. So fatty acid is where the name came from. So by the time you find these molecules floating around in water at pH 7.4, it would have a negative charge on this oxygen group here because the positive charge had floated away. Now this negative charge here, notices that these carbons on the glycerol have a partial positive charge, and so you can get these electrons here will bind on or will go on to this carbon. That forms a bond. Now that carbon has 5 covalent bonds. It doesn't like that, so it's somewhat unstable. The electrons between the carbon and the oxygen here will get kicked off, and so we have a hydroxide. When a hydroxyl group gets kicked off and it has a charge, we call it a hydroxide. And now we've got this hydrogen, or this proton, and the hydroxide, and the H plus and the OH, they can go together to make H2O. So we've pulled out a water molecule. Remember, that's called a condensation or dehydration reaction, and that means that we've now formed a covalent bond. So let's see. We have this oxygen here that had connected to the 3 carbon. I'm only drawing the 3 carbon here. I just have R above it because I kind of ran out of space, didn't plan this very well. But that oxygen that was right here is now connected to this carbon, and that's shown from this oxygen to this carbon here. That hydroxide group had been kicked off, so we don't have that anymore. Here's our hydrogen here and here. So now we've got the oxygen connected to this carbon. There still was a double bond to an oxygen here, so we have to draw that in. And so this is another functional group. It's called an ester bond, and it is formed when you connect something with a carboxyl group and a hydroxyl group. That's the type of bond that you form. So a carbon with a double bonded oxygen, and then another oxygen which is connecting the 2 carbons together. That's an ester bond. I'm bringing that up because we're going to see it again in a second when we talk about waxes. But before we get to waxes and steroids, I just want to take a minute to talk about the here's where I had written about trans fatty acids are not natural. They were thought to be healthy, but they weren't. So in nature, you're going to find the cis conformation far more often. And some of these fatty acids you might have heard of are omega-3 fatty acids and omega-6 fatty acids. Omega is a Greek letter with kind of the ohm sign there. The omega, or actually I should say, if you use you can use an omega to also mean ohm if you're dealing with electricity. But anyway, omega is a Greek letter. Alpha is another Greek letter. Alpha happens to be the first letter of the Greek alphabet. Omega is the final letter of the Greek alphabet. So here I drew a fatty acid, or well, the conjugate base of a fatty acid, because it doesn't have the hydrogen there. But anyway, so what I mean is that it's kicked off that proton, so we've got the negative charge. So it was a fatty acid, but it kicked off the protons, and now we've got conjugate base. And again, don't worry about that if you're not following along. We have the 1 carbon. This is obviously the most exciting end of this molecule with the carboxyl group, or the charged carboxide. I don't know, maybe that's what it's called. But anyway, we have learned that you start counting carbons from the most interesting end. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. My number's not very clear there, but there's 17. Now, most fatty acids in nature actually have carbons that are in multiples of 2. So I kind of messed up when I drew this. I just wasn't counting as I drew that out. So just know that we could have inserted one more zig or zag into that. But anyway, the main point here is that if you're talking about an omega-3 fatty acid, rather than counting from the alpha carbon, you count from the other end, which is the omega carbon. So here, the omega carbon, this carbon on the very end, would be considered carbon 1. This would be considered carbon 2 at this bend, and at this bend is carbon 3. So now we've got a double bond at that omega-3. So that's how omega-3 fatty acids get their name, is that the double bond is formed close to the end. And if we count from the end, the omega end, then it's the third one from the end, the third carbon from the end. The other one that we can talk about is omega-6 fatty acids. You might hear about that in nutritional advice. And again, I just redrew this molecule, but I put the double bond here. And so if we're, instead of counting from the alpha carbon, we count from the omega carbon. There's carbon 1, 2, 3, 4, 5, 6. So the double bond is here. That is an omega-6 fatty acid. And just because I'm focusing on this double bond doesn't mean there couldn't have been another double bond somewhere in here. I just drew them as simple as possible. Okay, so why do we hear about omega-3 fatty acids? It's because a lot of people, their diet, they have plenty of omega-6 in their diet, but they don't have a whole lot of omega-3. And it's important to have both because each of these types of fatty acids can be made into different molecules through metabolic pathways when you get into the body. Now, omega-3 fatty acids are precursors to lipid molecules that are anti-inflammatory. Okay, so in your immune system does a lot of talking with these proteins called cytokines and chemokines, but it also makes lipid mediators that can mean different things to an immune system. And so there are certain lipids that are omega-3, and they're precursors to the anti-inflammatory molecules. So if you have a diet that is rich in omega-6 and you're short on omega-3, one of the problems with omega-6s is that they can be turned into inflammatory molecules. And inflammatory molecules, there's an enzyme in one of those pathways called COX-2 that will turn omega-6 fatty acids into things like prostaglandins. I know I'm getting kind of in a deep dive here, but to bring this back to everyday relevance, if you take a Tylenol or an Advil or a Aleve as a pain reliever, you're trying to tamp down on inflammation. What you're doing is you're inhibiting an enzyme that uses this molecule, this omega-6 fatty acid, as a precursor. It stops it from making the prostaglandins, which are pro-inflammatory. So those prostaglandins are what can give you things like fever and such, and make you not feel well. So that's why Tylenol is considered a fever reducer, is it's blocking the conversion of omega-6 fatty acids into molecules that would give you the fever. Okay, and then our final slide for today, I just want to talk about the third category of lipids, which is a wax, and the fourth category of lipids, which is a steroid. And then here's cholesterol, which I accidentally said on that previous slide. So I just drew some fatty acid chains. They could have some double bonds in them, or it could be all single bonds. But the thing that I want to focus on is this group right here, where we've got a carbon, an oxygen, a carbon. So an oxygen is connecting two carbons together, and then this one of the carbons has a double bonded oxygen. And I'm going to sneak back a couple slides, and that is the situation what we had here, an oxygen connecting two carbons with a double bonded oxygen. So that was called an ester bond last time, and it's still called an ester bond this time. So a wax is a couple fatty acids connected together with an ester bond. Okay, so you've got waxes in nature. I mentioned earwax in lecture, but you also have bees make wax. Then you also have a lot of plants make wax for like protecting their leaves from dehydration. So you'll get a waxy layer built up on them. So that's waxes, and that's about all we're going to say about waxes this entire course. So take a picture, draw a nice picture, whatever. Steroids. The type of steroid that I drew down below is called cholesterol. So I really focused in on the fact that we have 6 carbons in a circle here, another 6 carbons in a circle here, and they're joined along this edge. Then we have another 6 carbon circle that is joined along this edge. And then finally we've got a 5 carbon ring. And at the tip of this 5 carbon ring is what is known as an R group. And the R group, as I drew it here, is what you would find on cholesterol. There's also a carbon side chain here, another carbon side chain here, and then a hydroxyl and a double bond here. So all of that fancy drawing will end up with a cholesterol. I just want to mention that as you go from one steroid to another, this R group is something that could be different from one steroid to the next. So there's where you could find some variation. And then over here we have an optional hydroxyl group, and any steroid that has a hydroxyl group there is known as a steroids with hydroxyls. Hydroxyls make alcohols, OLS, and here's the OLS, are sterols. So whenever this particular carbon on this group of 6 carbons has a hydroxyl group there, it's known as a sterol, which is a steroid with an alcohol group, a hydroxyl group right there. That's it for the lipids lecture. So go ahead and draw these out and be able to identify them. Waxes, again, 2 fatty acids with a ester bond. Steroids, these are so different than anything else that we've seen. If you ever see on a quiz, we've got this 6665 combination through here and some random stuff either here or not here. Those are steroids. So just be aware that whenever you see that, you should just automatically think steroid. Okay, that's it for today. So there's your second lecture that I posted today. That brings us up to speed. This lecture was first presented on January 26th. Again, this was Life in Biology. I'm Dr. Joel Graff, and you can feel free